Hey guys, I'm going to go over what we didn't get to today. So remember we talked about cell membranes and we did this bubble little activity to help us kind of model what a cell membrane does. So we had the challenge of getting the paperclip to go through the bubble, and you might have noticed how we have to coat the paperclip in the bubble solution to get it go through the membrane. And cell membranes are going to be very similar. So other features are cell membranes that they're fluid and flexible. They can self-repair. They can have compartments inside that compartmentalization, especially in eukaryotic cells specifically. There'll be proteins inside of membranes that have special functions like channels as shown here, as well as gap junctions, which are just the little connections between animal cells, and those connections are going to connect one cell to another cell. So another cell will be shown here. You can see the other cell, so very small but very short. And then we also have cells that can reproduce through binary fission, where they actually are having one cell and it splits into two. So these are all kind of functions of our cell membrane. We don't need to necessarily remember or memorize all of them, but just be aware of all of the functions and things that our cell membrane can do. Okay, other features of our plasma membrane or cell membrane are these proteins that I mentioned, like with that string that we had on the membrane. We have different proteins bound to the surface of our cell membrane, and these are called peripheral proteins. So they're going to be loosely bound to the surface of the membrane. So here we have this light, like yellowy color. That's the cell membrane itself. So we would have our hydrophobic tails in the middle, and our hydrophilic heads at the top and bottom. So these are proteins that are going to be bound to the surface of that membrane. Okay, and so think about, are these going to be hydrophobic or hydrophilic? You can pause the video and think about it. Okay, so these are going to be hydrophilic because they're on the outside where all the water is out here. They're going to be hydrophilic. Okay, so that means these proteins, think about what are the side groups or those R groups going to be of those amino acids. So they are going to be charged or polar, just like our water is polar. So that means our polar or hydrophilic region here will have proteins that are made of amino acids that are also hydrophilic and charged or polar. Okay, we can have integral proteins. So these are proteins that are interacting with the membrane, but instead of being on the outside like our peripheral proteins, they're going to span the whole cell membrane. Okay, and so think about now, are these going to be hydrophobic or hydrophilic? You can pause the video. These are going to be both. The majority of this center part will be hydrophobic, just like the hydrophobic tails, and then the ends will be hydrophilic, where they're interacting with the water on the outside. Okay, and so then think about what are the side groups that are going to be involved in these proteins. So in those hydrophilic regions, we're going to have charged or polar, and then we'll have non-polar R groups on those hydrophobic amino acids. So this is where that R group, remember, that's the tertiary structure that's being affected by those R groups, and so they're going to relate to the structure and how that protein interacts within the cell. Okay, these proteins are helpful in all sorts of ways. They provide for transport, cell recognition, so cells interacting with other cells, enzyme activity, signal transduction, which we'll talk about in, I think, Unit 4, intercellular joining, so joining different cells together, different attachment of things to the extracellular matrix or that cytoskeleton. So they have all sorts of functions. So we don't have to memorize this list, but just knowing that there's different functions for these proteins. Another aspect of our cell membrane is that it follows what is called the fluid mosaic model, or that's how we would describe it, is a fluid mosaic model. So think about, what's a mosaic? And from art, you might remember that mosaic is where we have different tiles and pieces that come together to make a picture. And the same idea is happening with our cell membrane. So it's the structure that's held together by those hydrophobic interactions, and they're weak. And so because of that, they're able to move and change and flow or shift. So here's kind of showing, and we can see these phospholipids all making up our cell membrane, and you see how they move, and that allows for the proteins as well inside of them to move as well. So that's what it's talking about as a fluid mosaic model. It's a mosaic because it's made up of many different pieces, and it's fluid because it's moving. Another important thing that is found inside of our cell membranes is cholesterol. So what is cholesterol? Cholesterol is a type of steroid. And so let's think about what type of macromolecules are steroids. Do we remember? These are lipids. And the reason a steroid is a lipid is because it is hydrophobic. So because of that, cholesterol is hydrophobic, just like our hydrophobic tails. It can be distributed throughout the membrane, and this is found in eukaryotic cells. And what it does is it actually helps to regulate the fluidity of the membrane under environmental conditions. So here we can see the purple phospholipids, and then there's some different proteins here in blue at different places. We have the integral protein, we have the peripheral proteins, and then we have cholesterol. And notice how cholesterol is inside of the hydrophobic region because it's a steroid, therefore it is a lipid and hydrophobic. And what it's doing is helping kind of keep that fluidity under control. We don't want the membrane to be so rigid and that it's unable to be flexible, and we also don't want it to be moving too quickly. So it helps to kind of buffer that fluidity of the membrane. Okay, there's also carbohydrates sometimes that are involved with the cell membrane. These are going to function as markers, and we have things called glycoproteins. That name is going to be related to what it's made of. Glyco, we can think like glycogen or glucose. So these are going to be glyco, meaning it's made of carbohydrates and protein. So it's like a carbohydrate protein hybrid. And so here's some examples of these glycoproteins that are attached here to peripheral proteins, and they're just acting as a signal for the cell for different processes. We can also have similar things called glycolipids, and so these are going to be carbohydrates that are attached to lipids. So that's a glycolipid. Okay, so we have these glycoproteins and these glycolipids. Again, these are going to serve as markers for our cell. So now let's think about what can easily go through the membrane. Okay, so are we going to have big things or small things going to be able to easily go through? We're going to have small things can easily pass through the membrane. Big things are going to need some help. What about polarity? Is it going to be nonpolar or polar? This is going to be our nonpolar or our hydrophobic molecules are going to be able to pass through the membrane very, very easily, like shown here, small nonpolar molecules. So those are the types of molecules that can move through very quickly. Examples of this are nitrogen gas, oxygen gas, and carbon dioxide. So these are very small molecules, and these all happen to be gases, but it doesn't have to be a gas that is the small nonpolar molecule that can pass through. Okay, everything else, like large things or hydrophilic things, they can't go just across the membrane super easily. They need help, and they're going to need the help of transport proteins. And these are these integral proteins that are going to serve as like a passageway for molecules to travel through that are large or that are hydrophilic. So it's not physically like moving and transporting the molecules, but it's more of a passageway that are these transport proteins. Okay, we can have channel proteins, like I mentioned, that are tunnel kind of types, and then we can have carrier proteins, and these similar function where it's like a channel, but it's more like a gate where it's only going to be targeted molecules allowed to go through. And some of these are going to require energy as well to happen, where only specific types of molecules can pass through. Okay, so what about those small polar molecules? Some things that are polar and very small can still pass through in some amounts. And these are, example is water. It's a small molecule, it is polar, and because of that, it's not staying in that hydrophobic region for very long, and so it's able to quickly pass through the membrane. We also have an issue of the cell wall being in the way in our plant cells. So it's like a structural boundary in those plant cells, and it's going to protect and maintain the shape of the cell. And it prevents the cell from rupturing or breaking apart when the water pressure is too high. And we'll talk about that when we talk about osmosis later in this unit. And it's going to help our plants to be able to stand up from the force of gravity. So, but then how does stuff get into the cell if there's this big cell wall in the way? And so it's able to do this because of a structure called the plasmodesmata. And plasmodesmata are small holes that are between plant cells that allow for the transfer of nutrients, waste, and other things such as ions. So here we can see we have a cell here, and it is a plant cell. We have in the grayish color is the cell wall, and you can see there are little gaps here. Those are the plasmodesmata that allows for molecules to pass through, and the cell membrane would be just inside of that. Okay, so just a review of what is the cell wall made of? Carbohydrates. Okay, so remember we have carbohydrates that are going to compose our cell wall, and different types of cells will have different types of carbohydrates making up that cell wall. So in plants, it is cellulose. In fungi, it is chitin. And in prokaryotes, it is peptidoglycan. So these are just different things, different types of carbohydrates that compose the cell wall in different types of cells. And again, peptidoglycan is a polymer consisting of sugar and amino acids. So glycan, we can think of glucose, and peptid, we can think of amino acids or our polypeptide with protein. Okay, that is it for our unit. Don't forget to do that FRQ practice. You just need to do number 1 and the graph for number 2. And then we have the quizzes on AP Classroom for this unit. There's also resources and other videos available on Canvas for this section. Bye, guys.